So cholesterol and triglycerides. Let's have a look at these issues here. So let's have a look what happens basically when we create energy from glucose. We know glycolysis produces pyruvate. Pyruvate then crosses the mitochondrial membranes into the mitochondria, and the substrate for the Krebs cycle here is acetyl-CoA. This spins around and produces our molecules of NADH and FADH2, and they go into the membrane, called the electron transport. We can also burn amino acids, as we've seen, but we can also burn fats. And we store fats as triglycerides. As I said, those triglycerides could be 2 molecules of palmitic and 1 of stearate, 3 palmitic, 2 stearates and 1 palmitic, and so on. They're just combinations of 3 fatty acids, usually carbon-16 and carbon-18, in other words, palmitic and stearic. Now, we need to then mobilize that to split it up into single fats again in order to start to then break those down, to oxidize them, to put them into the furnace as acetyl-CoA. So first of all, we have to mobilize the fat. Okay? So to split the triglyceride up, we need an enzyme called lipase. Just the same lipase that's produced in the pancreas and the small intestine to digest fats, we have to produce internally to break our fats down. So we have to have lipase. And lipase has to be sensitized by a particular hormone. Now we have 2 hormones that sensitize internal lipase. One of those is thyroxine, or the thyroid hormones, and the other one is adrenaline. So adrenaline will give you the quick breaking down of fats to give you the energy. So if you're in a stress situation, you will break down your fats much better. So a good way to lose weight is to get stressed. Okay, so you have a lot of stress and people can lose weight very quickly with a lot of stress. The other one is to increase your thyroid. So hyperthyroid people tend to lose weight. Hypothyroid tend to gain weight because they're not burning their fats. So what we've got here is hormone-sensitive lipase. So unless a person is under stress or not under stress, the regular breaking down of fats and producing energy from that is determined by thyroid hormones. So we need to just look at thyroid a little bit to see why that should be. Now, if we don't spin the Krebs cycle, and this is what we looked at in the last module, the spinning of the Krebs cycle, the speed of spinning the Krebs cycle, otherwise known as the metabolic rate, is determined by the level of T4 and T3, the biologically active forms of thyroid hormones. So if these are down, the metabolic rate slows down, which means we get a build-up of acetyl-CoA. Now we know yesterday we talked about acetyl-CoA being the substrate for going on to make the precursor to cholesterol and CoQ10. And if we block that at the HMG-CoA reductase enzyme with a statin drug, we reduce the production of cholesterol, but we also reduce the reduction of CoQ10. So we know that acetyl-CoA is the substrate for making cholesterol, and it goes on also to make more triglycerides. So this pathway is activated when the Krebs cycle slows down. So as we slow down, the more we put on cholesterol and the more we put on triglycerides. So to burn those triglycerides, we've got to increase our hormone-sensitive lipase. So either the adrenaline or the thyroxine hormones. So you see you go through this sort of vicious circle that you just go round and round and round with high triglycerides, high weight gain and high cholesterol, and a slow metabolic rate. So this burning of the fats is called beta oxidation and pops the residue of the fatty acid into the Krebs cycle as acetyl-CoA to burn as the fuel in the furnace. But if we don't spin it, we then just create more cholesterol and triglycerides again. So this is it on the chart of the energy. You've got this in your handout sheets here. So glucose, pyruvate, acetyl-CoA, and then that spins in the Krebs cycle. But if this doesn't move in the speed that it should do, it shunts it this way into making malonyl-CoA, which will then go on to make palmitic eventually, stearic, arachidic, and behenic. So any 3 of these form triglycerides, or like tripalmitates here, We then have to knock those out into the individual fatty acids with the addition of CoA, and then that CoA then breaks down two carbons at a time back into acetyl-CoA to feed back into the Krebs cycle. So this is why fat is a storage of energy, providing you can use it. You know, we put fat on for a rainy day, but if the rainy day doesn't come, then we just continue to have the fat on there. So this is looking at it in a little bit more detailed. So we've seen the spinning of the Krebs cycle, and this is a little part here where we go on to make our fats here, carbon 16, 18, 20, 22. Then any three of those are called triacylglycerols, such as 3 palmitates together, tripalmitate there. We then divide those up with hormone-dependent lipase into the individual, and then these go through the membrane, or the two membranes of the mitochondria, and they need a carrier. And this carrier to take it through, to start putting it into those, is called carnitine. It's called the carnitine shuffle. Sounds like a dance, really. So carnitine carries the fatty acid from the outside of the mitochondria to the inside, and then it just recycles again. So carnitine is a shuffler or carrier of the fatty acid from outside of the mitochondria to inside the mitochondria. So if you're deficient in carnitine, you may find again it's difficult to burn fat up. We looked yesterday at this pathway, again acetyl-CoA going on to make malonate and farnesyl pyrophosphate, and that was blocked at the HMDA-CoA enzyme here, which is what statins block. So as a result, we cut off our supply of CoQ10 and we reduce our cholesterol, which can be good if the cholesterol is too high, although we have to remember we get some cholesterol from our diet, but most cholesterol actually we make inside our body to the requirements. But if we make too much, then statins will tend to inhibit that, but at the expense of cutting out the production of CoQ10. So cholesterol, as we'll see, is a sterol-like compound, not to be confused with fatty acids. It's not a fatty acid structure, it's a sterol compound. And we break this down into hydroxycholesterol in the liver, and this requires vitamin B3, NADPH, oxygen because we're hydroxylating it, and vitamin C, which is what we said at the beginning that Linus Pauling discovered 50 years ago, and copper. So this hydroxylase enzyme uses copper as the electron transport. 7-alpha-hydroxycholesterol then goes into choline-CoA or choline-deoxy-CoA. So this is again needs B3 and B5 in this hydroxylase pathway. And those two compounds then conjugate with taurine or glycine, these amino acids, to form taurocholic acid and glycocholic acid, which are primary bile salts. So basically, when we get rid of or metabolize cholesterol, we turn it into bile. And so we've got bile salts here, tauro and glycochino-deoxycholics and taurocholics. So these with the 1R are what are called primary bile salts. And these further change to tauro-deoxycholic acid, which are secondary bile salts. So we have primary bile salts and secondary bile salts. So our bile salts are basically a series of conjugated cholesterol with taurine or glycine. So taurine and glycine are very clearly very, very important conjugators of cholesterol. So if people have too high cholesterol, this is where taurine and glycine may be important. So key nutrients in the metabolism or the breaking down of cholesterol: B3 in the form of NADH, copper, oxygen, vitamin C, taurine and glycine, and phosphatidylcholine, which carries old cholesterol from the cells to the liver. So phosphatidylcholine is a very important carrier molecule to take the oxidized cholesterol, in other words the cholesterol that's been damaged from free radicals, to the liver in order to convert it into bile salts. And we need selenium cysteine here, or selenium, which can help us break down, convert T4 into T3 to spin the Krebs cycle to speed up the metabolic rate there. So let's have a look at that again, as far as the thyroid hormones are concerned. So T4, T3 made from tyrosine. When we add one atom of iodine on, we make monoiodotyrosine, or MIT. When we add two on, we get diodotyrosine. And then when we add another two on, we get tetraiodothyronine. So we get two twos. So two of these molecules make 4, or tetraiodine, or T4, thyroxine. If we add a T2 to a T1, we get triiodotyrosine, or T3. Okay, so this is the way we would normally make T3, is a 2 and a 1, makes 3. Or we have a 4, which we take 1 away, the deiodinase enzyme, which occurs inside the mitochondria of the cells. So this is what happens in the thyroid gland. So we make T3, and we'll see in a minute, we also can make, in 40% of cases, reverse T3, which is like a blank. So let's have a look now. So again, now we can see the molecule, the tyrosine here. Iodine with diodotyrosine. Monoiodotyrosine just has 1 up here. And then tetraiodine, or thyroxine, has iodine, 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 iodine. And then T3, when we add 1 and 3 together here, gives you iodine, iodine, and iodine there. Okay. But if that iodine is in the other place, on the other molecule here, it's called reverse T3. And it's like a dummy, it doesn't work on the receptor sites. So you end up with a T4 level, but the reverse T3, the activated form, is like a dummy. So people get hypothyroid symptoms, although they have normal level of T4. So this depends upon being able to convert T4 into T3 into the cracked ones, which is the deiodinase enzyme. So here we see this thyroxine here converting into the deiodination to T3. 45% of deiodination goes into reverse T3 and to diodo, or T2, tyramine, and 20% is conjugated with glucuronate or sulfur in the liver. So this normal deiodination, in other words, making T4 into T3, in 35% of cases, goes through type 1 and type 2 deiodinase enzymes. So we actually have 3 variations on deiodinase enzymes, whereas type 1 and 3 would indicate that we push it more into the reverse T3, this pathway. So really the bad guy is an overexpression of the 3 deiodinase, type 3 deiodinase, which pushes it more into creating reverse T3. Now all these deiodinase enzymes are selenium dependent, and if you remember, it's the selenium in the form of selenium as an amino acid, usually as cysteine, where there's a gene that actually takes the sulfur out of cysteine and puts the selenium in instead. So this becomes called selenium cysteine. Now clinically, selenium cysteine very rarely seems to muscle test. But the one that does muscle test with this is selenium methionine. So selenium methionine seems to be the one which seems to where the insertion occurs. So when people show up to requiring, when you test for deiodinization, it tends to be that the people are not necessarily deficient in selenium, but they're deficient in getting the selenium incorporated by this gene in there. So it's a genetic polymorphism. So they have a polymorphism or a mismatch or poor expression of the deiodinase enzyme. And this is an indication they need selenium methionine. So sometimes you find the person will strengthen to selenium methionine and not to selenium itself, because they're not deficient in selenium, they just can't incorporate it. And this is a gene, and this gene is coded by U into G. U into G. So it means U, your uracil, goes into the G position because it's an RNA gene. So it's U into G. So that's the sort of combination. I'm going to put it on the next polymorphism charts that you get so you can remember. But put it in at the moment. If you find that you've got somebody who strengthens to selenium methionine, especially if they've got a deiodinase enzyme, and they only strengthen to methionine, selenium methionine, look for the polymorphism, which is U into G. Okay, so metabolic pathway, if you like, for looking at metabolism of cholesterol, because we always hear about cholesterol being bad and how do we lower it, how do we get rid of it. And the answer here is B3. Technically it's NADPH, but some people just use a high dose of B3 to do this, to stimulate this pathway, otherwise known as niacin. And as you know, niacin in high amounts can cause a vasodilation or a flush. If you've never experienced the flush, it's worthwhile. If you've never had a menopausal or any other flush, it's quite exhilarating really if you have one or two, but not as a long-term measure. So a niacin flush you can obtain by taking a high dose of niacin. So some companies have invented a non-flush niacin, which simply you can use niacinamide to do this. But some practitioners and patients actually like the flush because they say it opens up all their blood vessels and they feel better because they clear out all the debris in there. So we do an actual niacin or nicotinic acid as a supplement for those people who want to enjoy the flush itself. But for the rest of us, probably niacinamide will suffice. So B3 is niacinamide or niacin. Vitamin C is Linus Paulding suggested here. Copper, I personally never find, I can't remember the last time I prescribed copper, but maybe there are causes for it sometimes. If we need oxygen, we always think of adenosylcobalamin and iron. B5 for the CoA, but taurine and glycine as those conjugating amino acids may be very important here. Phosphatidylcholine as lecithin or omega oils, iodine and selenium for the thyroid gland. So they found a low ratio of CoQ10 to the low density lipoprotein or LDL, the so-called bad cholesterol, is a strong indicator of risk of atherosclerotic clogging of the arteries. So a low ratio of CoQ10 and low density lipoproteins. So natural sort of medicines in this, I put in sometimes substances which again can help lower cholesterol that are commercially available. Google, polycosanol, beta-sitosterol from plant steroids, cayenne pepper, pretty hot stuff, and garlic. These are traditional substances that can help lower down. We don't always know why a plant works medicinally. It may work by blocking, it may work by activating, opening up pathways. So we know that natural medicines, nutrients, work by opening pathways up by acting as cofactors and coenzymes, whereas generally pharmaceutical block. So they block the breakdown of things to build contents up. So it's done by blocking, which in other words poisons the enzyme or inhibits enzymes. Whereas what we try to do is to open the enzyme up and give it the cofactors and coenzymes. So this is more of an orthomolecular approach. So there's a little bit here about niacin, cayenne pepper, garlic. So some of the common remedies for lowering cholesterol. So now we've got the knowledge that cholesterol is largely produced as a result of a byproduct of not spinning the Krebs cycle properly, so look at the thyroid gland. So in all textbooks, biochemical textbooks, it tells you one of the symptoms or signs of a low thyroid function is high cholesterol. But nobody does it around the other way and says, oh, we've got high cholesterol, let's look at the thyroid gland. And most times it's not a low T4, it's a low T3. So it's the conversion which is the problem. Now, when we did the vitamin seminar on module 2, we found that vitamin D had a role here as well. That vitamin D is made from cholesterol, and if you're low in vitamin D, sometimes what the body will do is it will produce more cholesterol to put under the skin for you to convert a greater amount. So your cholesterol level builds up in your blood to deposit more there because you're vitamin D, with the hope that you get some sunshine and then it can convert that into vitamin D. So again, high cholesterol, think vitamin D, think thyroid and thyroid functions, and a sluggish Krebs cycle as a result. Now we're going to incorporate here homocystinemia. Again, we're going to talk about this in a future module, simply because it's involved with atherosclerosis, which is often the laying down of plaque of cholesterol and calcium. So we'll have a brief look at this. So S-adenosyl methionine, or SAM for short, is made from methionine, the amino acid, which is through a transfer enzyme here made into S-adenosyl methionine, which requires ATP, which then goes on to S-adenosyl homocysteine once it's given off its methyl donor. And then S-adenosyl homocysteine converts then to homocysteine itself, which will then go on to cystathionine and the amino acid cysteine. So this is called the sulfation pathway, transsulfation. At this stage with homocysteine, we can either recycle homocysteine by adding a methyl group from methylcobalamin, which gets its methyl group from methyl tetrahydrofolate and converts it back into methionine again. So this is called methionine synthase enzyme. Or we go on with homocysteine being reduced down to cysteine, which requires pyridoxal 5-phosphate or B6 and vitamin C in these two pathways. Now homocysteine therefore is a reactive intermediate compound. It's something en route to making cysteine, or it's the substrate for recycling homocysteine for this one carbon pathway, as it's called, to continue to make more methyl donors at S-adenosyl methionine. So it's a way of just making more methyl donors. So therefore, to recycle homocysteine, you need B12, methylcobalamin, and methyl tetrahydrofolate to recycle. On the other hand, to get rid of homocysteine, metabolize it through, you need B6 and vitamin C. Okay? So in cancer cases where we may have oncogenes, in other words, inherited genes which could promote tumors, we want to keep the caps on those genes. And those caps, to keep the caps on oncogenes, are methyl donors, methyl groups. So if we lose our methylation ability, you know, as we go low in our SAM, because we're low in our B12 and are low in our folates, we may release the caps a little bit or loosen those genes up, which may allow the oncogene to express itself. So this is why we have this pathway, to keep the methyl groups going. And this is why you find that the B12 and the folate are the most important methyl donors. So in a cancer case, we could actually have low homocysteine. So you may have low homocysteine, so you need that pathway to produce more homocysteine to recycle to make more methyl donors. On the other hand, in cardiovascular problems, as we see, you can have high homocysteine. And the danger of high homocysteine, as we see, is it breaks down collagen, which is what our artery and vein walls are made of, so we're more likely to hemorrhage and have cardiovascular problems. So high homocysteine would equate with cardiovascular disease, low homocysteine would equate with more malignancies. So procollagen is released into the extracellular space from the Golgi bodies in the cells, and this requires zinc and vitamin A. And here it's converted to collagen by peptidases enzymes and strengthened by cross-linking of the microfibrils by an enzyme called lysyl oxidase, which is a copper-dependent enzyme, which is inhibited, and this is the important thing, by high levels of homocysteine. So it means the final part of actually putting the microfibrils of the collagen, like the glue that holds us together, is inhibited by high levels of homocysteine. So the collagen is weak. And that weakness will lead to a stretching of the tissue, because collagen is what gives us the support. So we see that maybe in our cells, maybe in our patients, with a varicose vein. So a varicose vein is a typical homocysteine issue where you've lost the elasticity or the strength, the integrity of the vein wall, and so it's stretched and it hasn't bounced back out again. So a varicose vein is a typical example of high homocysteine, as are little red blood spots. So if you see anybody with little blood spots, or anybody has them, these are aneurysms or bulges in the little arterial blood vessel walls. So when you get a little bulge, you get that red blood spot. And if you got them there, you could have them here, you could have them here, you'd have them in there. And what you don't want is them to burst, of course. So we'll have a look at anybody who's got a blood spot in a minute. So the effect of homocysteine, you can read this, high homocysteine on coagulation. So ultimately there's two kinds of cerebral vascular accidents or strokes. There are two kinds of strokes. When I put this together, I thought, yeah, there's good luck and bad luck, isn't there? strokes of good luck and strokes of bad luck. And these are 2 strokes of bad luck. So there's an ischemic stroke which occurs when the blood supply to the brain is interrupted. So a cutting off of the blood supply, usually by a clot of some form, or an embolism as it's called, is the first type there. And these clots may be caused by hardening of the arteries in the carotid arteries, which feed the head and the brain with oxygen-rich blood. So any of these arteries in here could become blocked. So that will lead then to lack of blood and oxygen to the tissue distal to the clot. The second kind is a hemorrhagic stroke, which occurs when there's a bleed into the brain. And this is due to a weakening of the blood vessel wall. So this would be a hemorrhage into the tissue as a result of weakening of the small artery wall. So ischemic strokes are probably due to high oxidized cholesterol, in other words, the bad cholesterol, oxidized cholesterol. So omega-3 would be a good idea here to prevent aggregation of the platelets. Magnesium, ginger, garlic, onions to produce more synthesized, more prostacyclins from the prostaglandins 2 series. So ischemic, where we lack the blood supply. So we need to actually unblock the arteries, get the blood flowing again. So omega-3, and when omega-3 doesn't necessarily always mean fish oil. When we say fish oil, it might be DHA, it might be omega-3, 3, 6, and 9, or it could be just flaxseed in some people. Depends on, you need to muscle test it. And then to break the clotting down, we need to stimulate prostacyclins, so magnesium, and what we talked about earlier is GOG, or ginger, onions, and garlic. 3 very good food compounds to put regularly into the diet anyway, really. Ginger, onions, and garlic, 3 fabulous ones to keep the circulation going. Whereas hemorrhagic strokes are probably due to high homocysteine. Consider homocysteine factors, as I said, pyridoxal 5-phosphate and vitamin C. These are the main things we want to metabolize homocysteine rather than just recycling it, which would be your folates and your B12s. So here we see hydrogen peroxide in the presence of iron and copper going to hydroxyl radical, if you remember, which does a lot of damage. And hydrogen peroxide is produced in ischemic effects from superoxide up here and hypoxia above that. And this hydrogen peroxide will stimulate lipid peroxidation, which is what we talked about. So it's very important that we metabolize homocysteine and not allow that to go to metabolize hydrogen peroxide and not allow it to form the hydroxyl radical. So we don't want iron and copper available. So these people, iron and copper, of course, is made freely available from the red cells. When you puncture the blood vessels, the red cells ooze out, they die off and release the copper and the iron, and this adds to the inflammation effect of it. So the effect of a stroke is less to do with the actual hemorrhage locally, it's to do with the peroxidation of the tissue in the brain afterwards in the coming days, hours, and weeks. So the quicker you can reduce that inflammation after a stroke, the less damage you get. So even shoving aspirin in, any anti-inflammatory after a person's had a stroke will reduce the long-term effect. The quicker you can do it, the better it is all round. So by perhaps taking cloves, taking turmeric, anti-inflammatory, natural spices, garlic, etc. is a good idea. But certainly avoid taking multiple supplements which contain iron and copper in older age people. So most companies in multiple vitamin minerals do a multiple vitamin mineral without copper and iron, just in case a person gets a hemorrhage at any time, and then it stops that hydroxyl lipid peroxidation from occurring. So nutrients with ischemic cerebral vascular accident, omega-3, magnesium, ginger, garlic, onion, and selenium. With a hemorrhagic stroke, think P5P, vitamin C, and maybe the B12 folate, but it's mainly the P5P and the vitamin C. After the accident, what do you do after the accident to help the repair here? Patients we find often weaken to ammonia. Ammonia seems to be common, as a result of the destruction of the proteins and therefore the amino acids locally in the area. So when proteins are broken down, or deamination as it's called, they release ammonia. And if you can't get rid of ammonia, it builds up in the brain, it builds up in the kidney, and builds up in other tissues. So ammonia is highly neurotoxic, nephrotoxic, and it's even cardiac toxic. It can throw the normal heart rhythms. So to absorb ammonia, we need ornithine. So ornithine is something which is like a blotting paper to ammonia. We talked about that when we talked about amino acids. Magnesium citrate is important, or magnesium is important in the passageway of ammonia into citrulline. Biotin is also a cofactor in that enzyme. Vitamin C, zinc, phosphatidylcholine, P5P, NAD, and finally manganese, which converts the ammonia finally into the urea at the arginase pathway and allows us to weed out the ammonia. So let's just have a look at that urea cycle. You've got this in your handout sheets. This is important to learn that we produce ammonia normally by the breaking down of proteins called deamination of our amino acids, partly to put into the Krebs cycle to burn. So we recycle our proteins, and the removal of the amine part, or deamination, requires pyridoxal 5-phosphate. So this goes down to produce ammonia, and we also produce ammonia in several other biochemical pathways in the body. So together, this ammonia concentration builds up, and we normally produce then urea as a result of the ammonia in the liver. So all this actually occurs in the liver, not in the kidneys or anywhere else. Now we can produce ammonia also from enteric bacteria in the gut, and these would be primarily, as we'll see in the next module, gram-negative bacteria can produce ammonia, and parasites. So parasites can produce phenols, ammonias, and some other compounds. So they all add to the level of ammonia in a person. Now the part that you really need to learn is this. We've got ammonia and water coming together at this point here. So the first enzyme is carbamyl phosphate synthase enzyme here, which requires biotin and magnesium. And this enzyme basically converts bicarb and ammonia to citrulline. And as we can see, that really gives off carbamyl phosphate here, but is a biotin-dependent enzyme. Citrulline then goes around by arginosuccinate synthase to arginosuccinate, which then goes on to form arginine. And arginine is an amino acid, which is essential as far as building proteins, but can be formed in this urea cycle here, and will then give off the ammonia, 2 ammonias here, to form urea. If it doesn't go this through the arginase pathway, the ammonia will go on round and be just recycled. But this pathway also has a quick cut away from arginine back to citrulline with the release of nitric oxide. So this is the pathway where we produce nitric oxide, which is a powerful vasodilator of the blood vessels. This is why people sometimes take arginine in large doses to cause a vasodilation when they've got cardiovascular problems here. So you'll see that each of the enzymes here are all expressions of the genes, and so any individual polymorphisms within those genes can have a poor expression of the enzyme, and then you'll get a build-up of the substrate. So high levels of ammonia are very common post-stroke and also in many other conditions. So some of the symptoms you get is a tremor, slurred speech, blurred vision, joint pains, muscle pains, poor sleep, and most important is doom and gloom, thoughts of doom and gloom. Usually in the night and the early morning when people wake up, they get these horrible negative thoughts of doom and gloom, which lift during the day and then come back during the night again. And of course, during the night is liver time, isn't it? 1 till 3 o'clock. So this is the time usually they wake up in, you know, abject fear and things, and can't throw off these horrible feelings until they've urinated and got moving during the day. Stinging eyes because the ammonia can't get out in the usual channels, so it comes out in the tears, hot flushes, night sweats, impaired memory, cardiac arrhythmias, because of the cardiac, skin problems, chronic fatigue, pH imbalances, and muscle fasciculations or twitches. So it doesn't mean you get all of those with high ammonia, but you can certainly get enough of those to form, you know, real big problems.